Hey, good evening, everybody. Hello, hello. Hey, so welcome to tonight's Rock Talk. My name's, for those of you that haven't met yet, I'm Dave Buck, the Associate Director here at the lab. Thank you very much for coming tonight. So this is the second seminar series of your time here on the island. For those of you on the mainland joining us tonight, our seminar series, which we affectionately call Rock Talks, are sort of the opportunity for the whole island community to come together to learn a little bit about a particular special topic related to marine science, marine conservation. For those of you that don't know the lab or are new to the lab is jointly operated by the University of New Hampshire and Cornell University. We are quickly approaching our 60th year of operations, and we are proudly the largest and longest serving strictly undergraduate serving marine lab. So welcome to everyone. So tonight, we're really honored to have Heather Marshall with us, providing our Rock Talk. I had the great honor of meeting Heather in fall of 2017, I think it was, when sort of the brainchild of this shark biology and conservation class was formed. And so Heather's been here ever since, and I feel really fortunate to have her teaching this class for so long. So in the audience tonight, we've obviously got the shark biology and conservation class. In addition is the marine ecosystem research and management class. And then, of course, our long-running 10-week Scholl undergraduate research group. Hope y'all are here. Right on. So that's everybody that's here with us tonight. For those of you that are online, welcome. Thanks for joining us virtually. The format for tonight's talk is approximately 45 minutes. Heather will give a 45-minute seminar. It'll be followed with about a 15-minute Q&A. And for those of us here in the Commons, I'll walk around with a mic. And you can ask questions. For those of you online, please type your questions in the chat. And we have a couple of staff members that'll help sort of monitor the chat, and we'll get to your questions as quickly as we can. So it's a great honor to introduce Heather. Heather Marshall has a bachelor's degree from Smith College, a PhD from UMass Dartmouth. During her PhD, she studied a wide range of highly migratory fishes, particularly sharks, with a strong focus on physiology and particularly stress responses to capture. And a lot of her works look at sort of post-mortality following interactions with commercial fisheries. And she will talk a fair bit about that tonight. Following her PhD, she started a postdoc down at Mote Marine Lab down in Florida. And while she was in Florida, that postdoc actually took her all over the world, including work on salmon sharks in the North Pacific and a whole host of different shark species up and down the Pacific coast of South America, as well as, of course, here in the Gulf of Maine. She has a broad range of experience with multiple different species of sharks, ranging from white sharks, tiger sharks, hammerhead sharks. So we're really fortunate to have Heather providing us with our seminar. I should say one other thing. For many of you in the audience, you may already know this about Heather, but during all of that work with her master's or PhD and the postdoc at Mote, she also is the co-founder of the Gills Club. Raise your hand if you know about the Gills Club. Wow. Yeah. So, yeah, the Gills Club, and I may not explain this perfectly, but the Gills Club is an organization that really works to provide engagement opportunities for young female scientists to collaborate and work alongside other female scientists working in the marine sciences. So wonderful role model for all of our young emerging research scientists. Heather is currently a faculty member at the Ransom Everglades School in Florida. So she does a whole host of different things, and we're really fortunate to have her for this class and another round of shark conservation after this. So we have Heather on island for a whole month. Thank you so much for being here. It's a great honor to have you. Without further ado, please join me in welcoming Heather.
All right. Can you guys hear me okay? All right. I will preemptively apologize to everyone. My throat is a little hoarse from teaching for the last week and probably drinking too much coffee every day. But I will try best to keep my throat clear and project as well. So yeah, as Dave said, my background is in shark physiology, which means that I am super interested in how sharks' bodies work. I like to study their tissues, their cells. I think it's really fascinating about interspecific species differences. And so what we're going to focus on tonight is what happens to that physiology when sharks experience stressful events. So I'm going to focus on a lot of my research on a wide range of different species. And then at the end, I'm going to talk a little bit about the work that I'm doing with both high school and undergraduate students, including students here at Shoals. Oh. Oh. Really? Try to click method. There we go. Okay. So I always like to start here to kind of just have our broad view, right? What do we think of when we hear the word shark? And I know for a lot of people here within this room tonight, right, we've had a lot more consideration and perhaps appreciation of sharks out there within the oceans. But for the broad community, right, most people out there in the public, what they think of when they hear the word shark is a scary animal that is out there in the water, waiting to maybe interact with us when we get into the water. This is reinforced by movies. Of course, Jaws is kind of always spoken about in this context, and many, many subsequent movies since then that portray sharks in this way. And so we like to say that sharks kind of have a PR problem. Most people think that sharks out there are these big white sharks or big mako sharks that are trying to get at us when we're in the water. However, in reality, there are actually over 500 species of sharks that have been identified around the world, with new species that are still being discovered quite frequently. And as you can see, they have a wide range of body form, sizes. They are found in all different types of habitats, offshore, coastal, warm water, cold water, so on and so forth. And even our dwarf lantern shark there that you see in the middle is actually a full-grown shark. So a lot of shark species out there are actually quite small. So sharks definitely don't fit that reputation of being these very large species that are kind of nuisance. And for a long time, there was the reputation that the only dead shark or the only good shark is a dead shark. And so subsequently, our sharks have kind of not been studied as much as other vertebrates. And so as you might be aware, there have been many populations around the world that are experiencing population declines. So in terms of getting a global assessment of our sharks, this has been really challenging. So again, we've kind of been aware from a lot of good fisheries management over the years that local shark populations have been declining. But it wasn't until these studies started coming out, first in 2014, you see the top image there, and then with a follow-up study in 2021 that helped to explain the global status of our sharks. So these were our first global assessments. We're just going to go through a couple of take-home messages from these papers. First, it was determined that sharks are considered the worst reported extinction risk out of all other vertebrates. And these are some upper estimates within the paper, but just to say that it is thought that up to 200,000 sharks are killed every day around the world, and that 33% are threatened with extinction. Now, these are only the species that have been able to be assessed. There are many species out there that are still considered data deficient, and so we can't even make a call about what's going on with them around the world. Now, the usual culprits are at play here, so habitat destruction, climate change, pollution, but it's thought that these numbers are primarily due to overfishing. Okay, so when it comes to shark conservation, it's really incredible what we still don't know about our sharks out there in our oceans. We still have so many unanswered questions and really important pieces of data that are critical to conservation plans or fisheries management plans. Just going to highlight a few of them. They're listed there. So the first is basic life history information. So for many species, we still don't know how long they live, when they are at age for reproduction, how many pups they produce. So just that basic life history information that's so important. For many species, we also don't have a full handle on global fisheries impacts, which is what the rest of my talk is mostly going to focus on. So I'm going to come back to that one in a moment. In addition, though, we still don't understand their basics of migrations and movement. So for many species, we don't know where they are meeting to reproduce, where they are pupping, where nurseries may be, what are their seasonal migrations. So there's just an incredible amount of information that still remains to be discovered. If any of you out there are interested in pursuing shark research as a career, the story is not yet complete. So right now in this moment, I'm just going to focus on movement patterns. So in the last 15 to 20 years or so, our understanding of shark movement patterns, while there still remains a lot to be known, understanding where they go at certain times of year has greatly been facilitated by electronic tags. So electronic tags, just as the way they sound, they basically record data while they are deployed on the shark. So here we see an example of an electronic tag called the pop-up satellite archival tag. And while it is deployed on the sharks, it is recording depth, temperature, information about geolocation. It stores all that information. And then you can see in the graphic here, it will pop off of the shark at a predetermined moment that we program, transmit that data to satellite overhead, and then we get that data sent to our email, and we can start reconstructing what their movement pattern was. So this has really helped with migration patterns, particularly for those that are far-reaching migration patterns offshore, where these sharks are hard to follow and know what they're doing. So just as an example, when I was in graduate school, I had an opportunity to participate in a PSAT tagging project on juvenile porbeagle sharks in the Northwest Atlantic. We worked collaboratively with porbeagle commercial longline fishermen for this project, and we were able to tag about 20 porbeagle sharks for up to a year. So this type of data that highlights the movement patterns and the diving patterns and seasonal distribution and stuff like that for the juvenile porbeagle shark was something that we could then share with NOAA, fisheries managers, and help to fill in some of those data gaps. So these electronic tags have really been important in helping to move forward what we know about sharks, and we will return to them in a moment with regard to fisheries understanding as well. So now we're going to switch gears to focus on fisheries impacts and just talk about that for a moment. So as many of you in the room may be aware, sharks are targeted both in commercial and recreational fisheries. This depends on the species and whether or not they're considered prohibited or if they're being overfished, but they are targeted in specific types of fisheries. Commercial would be if they are going to be harvested for profit. Recreational is if they are being fished for fun. And so you have that level of fishing effort. And then on top of that, sharks are often caught as bycatch or incidental catch, which means that basically, fishermen will put a certain type of fishing gear into the water. So here we have an example of a long line gear. And we're going to talk a lot about long lines this evening. So I just want to make sure everyone knows what we're talking about. So a long line consists of basically a long main line, which you can see suspended by buoys there, close to the surface of the water. These can run for miles long, and suspended from the main line are going to be baited hooks that we call gangins. And so you'll let this gear out into the water and you let it sit for a period of time that's called soak time. And then essentially you haul the gear and you get whatever your catch is. So even if you are targeting something like swordfish, as you see in this image, these sharks love to come along, bite onto those baited hooks, and they become captured as bycatch. So there are several different estimates of this, but just to share one of them, it's estimated that sharks can be up to 90% of the total bycatch in some of these fisheries. And so the combined effect of targeted fishing and bycatch can become a problem for some of our shark populations and species. Now, if it is not a species that is allowed to be kept, or if it's the wrong time of year, or the shark is not of a particular size, a common management strategy is to enforce mandatory release, right? Kind of like catch and release fishing that we see in recreational fisheries. And the idea is that this individual is reported as an alive shark. It goes back to its population. It contributes to population growth. It's reproducing. And that basically goes into the reported catch for that shark. But this is based on the idea that these sharks are actually surviving the capture event. And what we've been learning through many, many years of highly collaborative research in many different facets is that all these sharks are not necessarily surviving the capture event. They may look good, they may not have any physical damage, they may be alive, but when you return the shark to the water, they can die hours or days later. And that is referred to as post-release mortality. So mortality that occurs after the capture event. So 10 or 15 to 20 years ago or so, there was a big push from NOAA and federal agencies to get a handle on species-specific, for certain species-specific levels of post-release mortality in particular fisheries. So how do we go about assessing delayed mortality? How do we know if a shark has actually died after capture, right? We're not viewing it. So one way that we can assess this is through tagging studies to calculate mortality. And another way that we can look at it is the physiological aspect. There are other methods beyond this, which I'm happy to share more about after the talk. But the first top method there with regard to tagging is what we're going to look at first. So this brings us back to those electronic tags. So like I mentioned, electronic tags will record information like water temperature and depth while they are deployed on the sharks. And there are two main types of electronic tags that I have worked with to assess post-release mortality, and they are commonly used in this type of research. The first one was that PSAT tag that I mentioned earlier, and you can see that shown ready to be deployed on a dusky shark that is about to be released. And then the next tag is in the lower image. That's called an accelerometer tag. And the accelerometer tag uses the same type of technology that we have in our phones that makes our screens flip when we turn our phone. So we can deploy accelerometer tags on these sharks and actually look at their physical body motion while the tag is on the animal. The man there to the left is my postdoc mentor, Dr. Nick Whitney. He has designed many different types of tag packages for deploying accelerometer tags. You can see him there getting one ready to go on a white shark. So the piece of information that we mainly focus in on is depth for these studies. So sharks' bodies are negatively buoyant, which means when they die, their bodies will sink. And so in the image you see there on the right, which is from blue sharks tagged by Chris Moyes, on the y-axis there you see depth, and on the x is time after tag and release. So essentially this shark is released from the boat. You can see it swimming along, and then you see this sinking pattern. So this essentially means that the shark has experienced a mortality event. Its body is sinking, and this shark happened to be in very deep water. So you see it go quite deep, and then at a certain depth, that tag will pop off of the shark, shoot up to the surface, and transmit that data to overhead satellites. If the shark is swimming in a shallower area, you'll just see that body sink and sit on the bottom. And particularly for species of sharks that need to keep swimming in order to breathe, we would not expect to see this type of behavior. So we determine post-release mortality by visually inspecting the depth behavior. So just as an example of one of these projects, so this was my dissertation work at UMass Dartmouth, where we were assessing mortality rates of the sandbar and dusky shark in commercial longline fisheries. You'll see there the term at vessel and post-release mortality. So at vessel is essentially the percentage of individuals that come up as mortalities at the time of capture. So we wanted to run a long line survey where we could focus in on those at vessel and post-release mortality rates. To be as realistic as possible, we mimicked the fishery gear specifications as closely as we could, and we ran a long line survey in collaboration with Virginia Institute of Marine Science. We did make one gear modification in that we integrated something called hook timers into the Gangens for this project. So if you look up in the upper right-hand corner, you'll see our Gangens there, those baited hooks that are baited and ready to be deployed on the main line of the long line. At the top, you'll see the tuna clip, and then you see those dark cylinders. Those are the hook timers, and then below are the baited hooks. So over here, we see a close-up image of how these hook timers are integrated into the gear and how they work. So essentially, you have a magnetized stopper that is part of the hook timer, and then you're going to have a line that goes to the hook. So if a shark comes along, takes the bait, starts thrashing around, it's going to pull out that magnetized stopper, and that timer will start recording and record continuously until we haul the gear. That's at least the idea. It doesn't always work perfectly, but for the most part, that's how we get our data about how long each individual shark was actually on the line for. So if you remember, we mentioned soak time, right, which is how long the long line is in the water for. And in commercial fisheries, that ranges from about 12 to 24 hours. But this allows us to focus in on shorter periods of time to see if there's a threshold above which we start seeing significant mortality. So just to go briefly through the results for the project, we were able to assess post-release mortality rates for the sandbar and dusky sharks using those PSAT tags. So for the dusky shark, that came in at 29% for our study, and for the sandbar shark, that was at 20%. So we are seeing a degree of sharks who are experiencing mortality after capture. But in terms of using those hook timers and looking at thresholds of time, what we found is that for sharks that were on the line for greater than 3 hours, when you combine their at-vessel mortality rate and post-release mortality rate, so total mortality, the situation painted a slightly different picture. So for sandbar sharks, that overall mortality rate was 47% for sharks that are on the line for greater than 3 hours. And unfortunately for the dusky sharks, it was closer to 97%. So obviously extremely high associated mortality rates for this gear. What is concerning is obviously the long period of time that these sharks are typically on the line for. But also what's interesting here is the species-specific differences that we're seeing. So sandbar and dusky sharks, as you can see with their scientific names, they are congenerics. They have the same genus. They're found in very similar types of bodies or bodies of water. They're very similar sharks. But why do we see one species that is so much more sensitive and succumbing to that capture relative to others? And this is going to be a running theme throughout the rest of the talk. For my postdoc in Florida, we conducted a several-year project working collaboratively with commercial captains in the longline fishery in mostly the Gulf of Mexico. We were able to tag a wide variety of different species. The fishermen did all of the fishing, and we were there just to put on tags and take blood samples. The tags we use for these studies are accelerometer tags, which record a huge amount of data, but they have to be recovered in order to get that data. And so, but the benefit of that is that once you recover them, they can actually be reused again over and over and over again. So it's a much more cost-effective way to get really nice high sample sizes. So this figure here, I know it's busy and it's a little far away. I'll walk you through it, but basically this is a table from this project. So you can see that we sampled sandbar, blacktip, tiger, spinner, bull, and black-nosed sharks. Using our collaborative method and with the accelerometer tags, we caught over 900 sharks. We tagged over 300 individuals, and this allowed us to have a really nice sample size for these species to look at the total mortality rate when exposed to longline gear. So again, the take-home message here are these interspecific differences. So if you look at the, let's see if I look here, the bull shark, right, with a 7% mortality, or the tiger shark with a 2% mortality, right? Those are pretty low. But if you compare that to blacktip with 62% or spinner with 89%, again, these are all very similar species that are, well, they're not similar species, but they're found within the same bodies of water, and they are all being exposed to the same types of gear. So why do we see these species-specific differences? And so that's where we dive a little deeper, and we maybe look to the stress physiology of it all. What's happening internally to cause mortality within these different species? So we're going to turn to our little stress physiology primer now. So like I said, physiology is basically the study of cellular and tissue processes that maintain homeostasis within these animals. And so stress physiology is a cascade of changes to the typical cellular processes when they experience a stress event. And there are many other scientists who study different types of stress physiology that are not necessarily fisheries related. I'm just going to be focusing on those fisheries-induced stress physiology. And how do we understand what's going on internally? Well, we take a blood sample. So just like you or I would go to a doctor to assess our health through a blood sample, we can do that with our sharks and look at certain parameters that are indicators of the stress response. So the model of stress physiology is a 3-tiered response. So this is kind of like what we expect to happen within our sharks based on what we know about stress response in general, many other vertebrates. A lot of the pathways are similar to what happens to us when we experience a stressful event. So you're going to have primary, secondary, and tertiary levels of the stress response. The primary will be initially when the shark detects that it's been captured, and it's going to start kind of having that fight or flight response. What triggers that is a hormone release, basically to help the shark try to get away and recover from the stress event. That's going to lead into the secondary, which is a variety of biochemical, metabolic, and molecular responses to that hormone release. And again, initially, this naturally happens because it's meant to help the animal. It's meant to, help fuel their muscles while they're thrashing on the line, help them recover from the stressful event. But what becomes a problem is if the stress event lasts for a long enough period of time, normal cellular processes can start to break down because of the exacerbation at the secondary level. And then the tertiary response is essentially the effect of stress carried over to population level. So let's say, for example, a shark experiences a stressful event and has its ability to reproduce affected. That would be a tertiary response. So in terms of what we study within our sharks, we primarily, at this point in the state of the research, focus on the secondary response. So at the tertiary level, I'm sure you can imagine that's incredibly hard to study. Some scientists are starting to do some work, but that's going to be, long-term, large-scale projects to understand. The primary level, usually people think of the stress hormone cortisol, right? That's kind of what we think of when we hear about stress. Sharks don't have cortisol. They have a similar molecule called 1-alpha hydroxycorticosterone, and I can spell that for you later if you want to write it down. But basically, it's a different molecule that serves a similar role to cortisol, and we simply just don't have the biotechnology methods to efficiently study that. They haven't been developed. There are scientists right now who think they have good assays working and are working to validate their assay for that hormone. But most of the work up until now has really focused on the secondary parameters. So what do we expect to see? When a shark initially detects that it has been captured on the line, it's going to experience that initial primary response, the hormone release. That's going to trigger the fight or flight response. It's going to start thrashing around. There's going to be increased muscle activity. At the same time, it's hooked. It's not going to be able to swim the way it normally can. And so all of the important oxygen and CO2 exchange that happens at the gills, along with ion exchange, that can be affected by the shark being physically captured and not able to swim the way it normally would. So what we can expect to see there are carbon dioxide increases within our blood, as well as ion fluctuations. Because of the increased muscle activity and metabolism that kind of drives that increased muscle activity, there's going to be switching over to that anaerobic metabolic pathway, which the byproduct is lactic acid. Again, the same type of metabolic pathways that we have. And so we can expect to see increases in blood lactate and also potentially increases in blood glucose. And the glucose would basically become available to those swimming muscles to fuel those swimming muscles as the shark is thrashing and trying to get away. Now, with the increases of lactic acid, as well as the increases in carbon dioxide, both of those will cause overall blood acidosis, and we can expect pH declines within the blood as well. So these are all the parameters that we look at when trying to assess the stress physiology of our sharks. Okay, so again, busy table. I will walk you through it. I just want to highlight a couple things about it. But essentially, when we are kind of setting out to do a stress physiology of sharks in a particular fishery, the first thing you do is just kind of take that blood sample when they come up on board, and you get that snapshot of what the blood looks like at the time of capture. So when I was in graduate school, the lab that I was in, we were all going out on long line surveys all the time, and we would just simply blood sample any species that came up. So it gave us a nice data set of these 12 different species after long line capture. And so you can see the secondary parameters that we look at: sodium, calcium, chloride, potassium, magnesium, and then we look at glucose and lactate as well. So again, this is just kind of how these projects move forward. First, you get this snapshot of blood. And just a couple of things that I want to highlight here. For example, if you look over in the lactate column, if we look at something like the blue shark with 4.8 millimolar of lactate, or the oceanic whitetip with 0.3, right? Those are pretty low lactate values. A baseline unstressed parameter is in the range of 0.5 to 1.5 millimole of lactate. So those are pretty low. But if we compare that to something like the silky shark with 16, or the mako up to 16 and sometimes getting up into the 30s, again, we're seeing very distinct species-specific differences in how they are responding to the longline gear. So again, all of these sharks were on longline surveys. We didn't know how long each individual shark was on the line for. We just got that snapshot of blood when they come up on the boat. But getting back to the concept of using hook timers, if we want to kind of look at the time frame where this stress response is unfolding and maybe look for areas where the blood is becoming more and more exacerbated, we can use those hook timers to basically pair up individual blood values with how long they were on the line for. So this is getting back to that sandbar and dusky shark work. So in the top are our dusky shark figures. On the bottom are the sandbar shark figures. On the left are both potassium. On the right are both lactate. And these are like linear regression analysis. They're called gamma analysis. It basically allows us to look at the curve of the data so we can see the shape of what's going on with these blood parameters as the sharks are on the line for a longer and longer period of time. So each individual point you see there is one shark, how long it was on the line, and what its blood looked like. So this allows us to, again, kind of see what's happening with longer timeframes of capture. So other scientists do similar types of timeframe work. So for example, this is a colleague and friend, Dr. Jeff Kneebone, who did his PhD work looking at sand tigers after capture on recreational gear. So again, on the x-axis, you see angling time in minutes, and all of the figures are various blood parameters that we can look at. And again, you see just in a short period of time, those blood parameter values are starting to increase. So again, for each species, we're just trying to get a handle on what's happening physiologically when they are captured. So that's how we look and try to assess whether or not they are stressed. But can we understand what's actually causing mortality from the stressful event that they are experiencing? What's driving those rates of post-release mortality? So this is where we combine our tag data with our blood data. I'm going to show you a series of figures. They're going to look intense at first, but I promise I'm going to walk you through them. But essentially what we're looking at here are carbon dioxide values for each individual shark that we captured in that Florida longline work where we were working with the commercial longline fisheries. On the left are species-specific at-vessel mortality percentages that we calculated for that project. On the right are species-specific post-release mortality values. So basically for each individual, we were graphing their species-specific mortality rate with their blood value. The red circles are individuals that experience post-release mortality, gray are at-vessel mortality, and the black circles are those that survived. So just keep in mind, gray and red are mortality events, black circles are survivors. So in this way, we start to see what's going on with the blood, where we start to see higher and higher rates of mortality. So we can do this across a range of parameters. So here's carbon dioxide, here's the same exact figure structure for lactate. So there, even with those higher mortality values, we're still seeing not necessarily lactate values that are super high, right? Some variability there. So maybe they're stressed, but maybe lactate isn't necessarily causing mortality, right? So there's a difference between stress and causing mortality. With pH values, we expect to see those declines, which is what we see there as well, with increasing mortality events as well. One parameter we always like to talk about is potassium. So across a lot of these studies, we start seeing potassium showing up over and over again as a possible predictor of mortality, or I shouldn't say predictor, correlated with mortality and potential to predict mortality if we understand what's going on better. So these are the same types of figures that I just showed you, and I'm just going to throw a threshold on there of 7 millimole blood potassium. So if we look above that line, right, we see a lot of those gray and red circles, a lot of mortality events. Below that line, a lot more black circles. So there seems to be a threshold here. And if you go to the blood potassium literature, what you'll find is that 7 millimole is a threshold which is referred to as hyperkalemia in mammals. So if mammals experience hyperkalemia and that threshold value is 7 millimolar, they can start to experience cardiac upset, which can lead to mortality. Specifically, they experience bradycardia or slowing of the heart. If you remember intro bio and the sodium potassium ATPase pump, potassium is a really important intracellular ion that also plays a huge function in muscle contraction physiology. So if our sharks are experiencing their own type of hyperkalemia, where maybe these elevated potassium levels are impacting heart function or impacting the ability of their swimming muscles to function properly, right? That will limit their ability to recover after the capture event. And when we release them, we think they're going to go on to survive, but maybe this physiological upset is affecting their heart and affecting their ability to swim after the capture event. So for the work that we did in Florida, we did some, these are referred to as decision tree analysis, where essentially in a species-specific way, we take a snapshot of what the blood profile looks like and compare that to those individuals that were survivors and those that were experiencing mortality. So for example, in our sandbar sharks, for those individuals that have potassium values less than 6, we're seeing 0% post-release mortality. But if their blood values are higher than 6 and combined with a pH value less than 7.2, then we start to see a much higher rate of post-release mortality. For the spinner shark, lactates seem to be really important in terms of correlating with mortality. So the reason we do this decision tree analysis is it raises the question, can we take a blood sample from a shark and based on that species-specific profile and a lot of modeling and a much better understanding of what's going on, can we predict mortality from the blood sample alone? So the reason we may want to do this is sometimes tagging to determine post-release mortality is just not feasible for the area you're working, the species you're working with. These tags are incredibly expensive. The PSAT tags I showed you are $4,000 a piece. These studies are incredibly expensive. So if you can go in, get a blood sample, and again, we develop a much better understanding of what's going on, perhaps we can use that predictive power to understand possible outcomes when tagging is not a possibility. So to start rounding things out, the question of can physiology aid in mitigation? So the most important thing here, in my opinion, or one of the most important things, is understanding that species-specific difference, right? Not every shark will react the same way to the same type of gear. And so they need to be considered differently in terms of how they're handled, how they are fished for, so on and so forth. Possible mitigation measures. So is it possible to reduce soak times in commercial longline fisheries? That's kind of a question, kind of getting back to that idea of thresholds of time. How long is too long for certain species? Are there potential gear modifications that will aid sharks in being able to swim better and maybe start recovering while they are still captured on a line? Highlighting the importance, particularly with recreational fisheries, of keeping the gills wet, keeping sharks in the water, helping them recover while they are being released, and even some resuscitation, right? Putting a hose in the shark's mouth with a saltwater flow through will allow them to breathe and start recovering while they are basically being released. The effects of potassium need to be investigated. So if we understand better how potassium could potentially be impacting the heart, is there something that we can do to help promote recovery? And then I just mentioned that idea of predicting mortality based on our blood sample, which we have a lot more work to do before we get there, but we are starting to see some of these blood parameters maybe having that power. So stress physiology folds in under this heading of conservation physiology. So if any of you out there are interested in how the bodies and the cells and the tissues work, but also really want to contribute to conservation, this is a nice place to land. It's basically using what we understand about physiology in the aid of conservation. And at least within the field of shark physiology, it's a relatively new area. This is a journal that was first created in 2013 by the name Conservation Physiology. It's open access if any of you are interested in this type of work. It's not just sharks or fish, it's any scientist out there who is using physiology to try to aid in conservation questions. And in the last 10 or so years, there have been a number of papers that have come out highlighting the idea of where physiology fits in to the grand scheme of, say, fisheries management in collaboration with fisheries, the management and policy to promote best handling practices and promote survival for some of our species. So as an example, how does this work carry over into policy? So while we were doing the work in Florida on all of those other species, we were also tagging and blood sampling scallop and great hammerhead sharks. Scallop and great hammerhead sharks are both critically endangered species that have experienced overfishing in the past. And so several years ago, the Southeast Data Assessment and Review Cooperative Group, which is under NOAA, put out a call for any kind of fisheries related data for these species as they began a stock assessment and review how to best help these sharks moving into the future. And so even though at the time we didn't have these papers published, we were able to submit our data as working documents so that they had that data just for consideration if it could help within that assessment process. So we do try to get this data directly into the hands of the people who are making decisions, just to add information as they go through these stock assessments. Moving forward, so like I said, we're kind of at the tip of the iceberg here, looking at those secondary stress parameters. My research now is kind of moving into other areas that might also contribute to the state of the knowledge. So are there other things that we're not considering? So I'm interested in using molecular tools to look at the gene expression of important proteins in the stress response pathway. Also getting back to that primary response. So I've sent myself and a bunch of collaborators have sent a bunch of blood samples to the scientists who are developing that assay for the 1-alpha hormone, just hoping to help them as they develop these methods. Also, the mechanisms of stress and recovery are really interesting to me, right? We kind of have an idea of what's going on just based on the basics of physiology, but the image you're looking at here are all of those little protein exchangers that exist along a cell membrane. They are moving ions and protons back and forth, just to maintain homeostasis, but also in response to stress events. So we really don't know what are the actual mechanisms at a cellular level of recovery. So I think that's super interesting. And then, of course, excuse me, the species-specific differences, right? We really don't have a good handle on this. We're seeing it very clearly in the physiology and the mortality. It's likely a combination of physiology and behavior. There are some other theories out there, but a lot more information remains to be discovered here. So moving forward, the state of my current career right now not only is trying to move into these other questions, but also involves a lot of shark-related education. So I am pursuing a lot of these projects in collaboration with both high school and undergraduate students. So I just wanted to talk a little bit about those projects, and then we will wrap up. So as Dave mentioned, I'm a co-founder of the Gills Club. This was my first experience with shark-related education. And basically, we created Gills Club because we saw the need. We were meeting young girls at public events who were being told at school that sharks are only for boys. And obviously, if you've ever watched Shark Week, there's a real devoid of women on those boats. In reality, there's actually a lot of amazing female shark researchers around the world who rarely get featured in programs. And so we wanted to connect girls who are interested in sharks to these women and help girls get experience on the boat, on the water, learning about sharks, learning about marine science. And then, of course, this brings me to our course here at Shoals Marine Laboratory, Shark Biology and Conservation, which I've taught for the last several years with dear friend and colleague Maggie Winchester. And of course, this year we are very fortunate to have Jake Jerome here as well, contributing to the class. Just a shout out to how collaborative the development of this course has been. Up in the right-hand corner there, you see the prototype of the shark cradle that we have now, which made everything that we do possible. And obviously working closely with Captain Steve in the middle there, who you can see handing off a baby mako shark to a very happy Maggie in one of our fishing days. And then, of course, down here, basically everything we do with these sharks is a collaboration between faculty, students, and staff at Shoals. Makes it all possible. And so we also collaborate with our high school sharks class teacher, Dr. Ashley Storr, and all of us are basically working to create this kind of long-term data set, right, around fishing effort, what we're capturing, and of course, the stress side of things. So during our course, students are learning the methods of blood sampling, processing. They are handing the blood samples. All of our students today on board learned how to blood sample. Mackerel, not sharks, but that's the beginning. We've also done other fun projects here at Shoals where students are getting hands-on experiences, always highlighting the incredibly important job of data collection. Our students there in the lower right-hand corner are taking a little nap between data collection on the boat. But again, all of this is kind of building towards this long-term collection of projects as we move forward. So in Florida, where I live, again, I'm moving forward with these molecular questions. In my previous position at State College of Florida, we really worked a lot on developing the quantitative PCR method for looking at expression of proteins. And then in my current position at Ransom Everglades School in Miami, we have an after-school research program. This is a high school, so the school is very supportive of experiential learning. And within our YREP program, which stands for Young Researchers Program, we do shark research, and there's also a physics teacher who does laser research. So our students created the logo with a shark with a laser on its head. And so we are doing a lot of collaborative work. The next stage of that is trying to use these lasers to potentially look at contaminants in shark tissue. We also developed a biology and ecology sharks course down at Ransom, where our high school students are also getting shark research experience, fishing, handling sharks. They're collecting data and contributing to our projects down there. And one project that we are particularly excited up here that we started at Shoals a couple of years ago is microbiome sampling of our sharks. So looking at the microbiome communities that live on different shark species. A lot of the methods were developed by our TA at the time, Liz Whitson, who's swabbing a little baby cat shark there. But then, of course, when we're out in the field, our students are learning sterile techniques, aseptic techniques to get our microbiome samples from our sharks. And then this project is kind of linked to work that we are now doing at Ransom in Florida. This past year, we had a senior who did an independent study where he learned all of the methods that we had developed up here and then helped to move the lab work side of it forward, working with processing of samples, DNA extractions, and stuff like that. So it's really fun to have the project going in both areas. So of course, with all of this work, I hope that I am supporting dozens of future shark scientists out there who are going to put their own stamp on the field. But of course, not everyone will necessarily move into that field, but for future vets, future doctors, just future shark advocates, to give them that hands-on experience is really exciting. And they are contributing to moving this work forward. So as we wrap up, I just want to give a small plug if anyone is interested in learning more about the Gills Club. It is the education initiative of the Atlantic White Shark Conservancy in Massachusetts. Our logos are there, and we also have a podcast called Gills Club if you want to check that out as well. With that, I will happily take any questions. Thank you. Should I leave this on? Okay.
Yes, please. I'll
Okay.
just keep a distance to him.
Awesome.
Thank you so much, Heather. That was a wonderful talk. I love hearing
Thank
about
you.
some of the work that's also happening in Florida.
Thank
All right, questions. Must be some questions.
Disclaimer, I am woefully uneducated about sharks, but I know for like seals, for entanglements, they have like seal rescue teams and like seal rehab centers. Do you think something like that would be realistic for sharks?
I don't think necessarily it would be realistic. Nothing, I don't think anything like that has ever been attempted unless it is a shark that is being held at like an aquarium for transport to another aquarium or something like that. The transport process can be pretty stressful for many species. Typically, the thought is, if there is a shark that is appearing to recover, what we'll do at boatside is kind of like, swim the shark back and forth, try to get water moving over its gills, try to resuscitate. In some labs, shark scientists will actually get into the water with the shark and swim it down and stuff like that. But obviously that's in a research setting, not necessarily a fisheries setting. I think it's more about kind of getting best practices in terms of handling, right? Keeping the shark in the water as long as possible. Some research groups will do like a little like nutrient injection before they release the sharks. So there is talk of like maybe there's some sort of an injection that could be given to counteract something like hyperkalemia. But, these are kind of theoretical and definitely would have to be like heavily tested out. But generally speaking, the thought is to just get that shark back in the water. As long as it's swimming, it hopefully will recover. Yeah, great question.
Thank you so much for talking again. I was just wondering, the intersection of physiology and conservation was like really, really interesting and
Yeah.
sounded like really unique to me. Can you like explain a little bit more about that? Maybe not just like in terms of like shark stuff, but like I know you mentioned there's a certain scientific journal. So any other examples you know of in a field? Because I just thought it was really interesting.
Yeah, absolutely. So just like generally speaking, I mean, yeah, and so other fish species, there will be similar types of studies like fisheries capture, looking at, different birds and stress that they may experience due to, influences on their migration pattern, or a big one that's, bubbling up, right? Climate change, so if we understand physiologically what's going on, and then there's something like ocean temperatures changing, applying that knowledge of physiology to theoretically describing what could happen. Yeah, there's so many different areas where, taking the knowledge of just how the body is working can be applied to any kind of anthropogenic situation. So that also can include habitat destruction, pollution, So yeah, anything that would raise like a conservation challenge, maybe there's some knowledge of physiology that can help with understanding that. Yeah, so definitely check out the journey. It's open access. There's all types of stuff in there. Yeah.
Thank you for your talk. I was wondering for your studies on the, I guess, post-release mortality, how you chose which species to look at and also where to do those studies, since you knew there was potential for mortality. Like you determine like certain species or areas where the sharks weren't, like the populations were more thriving.
Yeah, absolutely. Yeah. So for the sandbar and dusky shark, that was definitely a very an example of a very specific need within the shark fisheries management. So the sandbar and dusky shark had both been historically part of the commercial longline fishery, and then through stock assessments, it was determined that they both were experiencing overfishing. And so actually, within the grand scheme of things, their fisheries were shut down. So you were no longer allowed to target sandbar and dusky sharks. And if you caught them, you needed to release them. At the same time, they actually established a research fishery for the sandbar shark. So this was like a small group of selected boat captains who were allowed to still longline fish for sandbar sharks in collaboration with scientists, with the idea that there's data sharing and trying to get a handle on fisheries impacts. So that sandbar research fishery remained, and then just from other generalized bycatch exposure, both sharks were still being caught, even though their fisheries were shut down, both sharks were still being caught as bycatch. So the situation was much more dire for duskies than sandbars, but basically there's, you know, when NOAA puts out like funding cycles, they will list things as priorities. And around that time, the priority was getting those post-release mortality rates specifically, especially for the dusky shark, but also the sandbar. And then in location, we just knew that we could kind of get a readily accessible collection of both species at the same time off of the eastern shore of Virginia. So the Virginia Institute of Marine Science has an eastern shore lab, and we would go, 20 or 30 minutes offshore and set long lines, and we could readily find both species. So it allowed us to really hone in on them. In the Florida fishery, again, they were just like various species that, were being called as priorities to investigate. And so we started out, I can't remember now, when we first started doing the project evolved for many years, but essentially at a certain point, we were just kind of tagging and sampling most things that came up, so that because we saw the opportunity to sample all of these different species, because we were capturing them and we were using accelerometer tags, and so we could reuse these tags and just push up our sample sizes. Towards the end of that project, hammerhead sharks really started becoming more of a relevant species, and so we really started going out on just hammerhead specific studies. And we were working with our commercial captains. So in terms of finding them, we just communicated with them. I mean, they know those waters way better than we do. They fish those waters. And so they were able to say, this is a good spot for this and that. And then we would tag what was coming up. Yeah, great question.
You may have answered this already, and I know you mentioned the injections, but I'm just curious how you guys manage the stress response or like any mortality rates for sharks that you are catching for like blood sampling or research,
Yeah.
or
great
like if you're just careful to work within the threshold, or how you deal with that.
Yeah, that's a really great question. So again, just like highlighting those species specific differences is kind of like the first step, really, really important, right? So if we're out tagging sharks, not for a mortality study, just for something else, we know that if we get a hammerhead shark, we need to be super fast. Like that's a sensitive couple of species. And so we're going to work through that process in a much more streamlined thing. Maybe we'll cut out some things that we sample. That's going to be like as quick as possible. Whereas if we get something like a tiger shark, we're still going to be as quick as possible, but we know that they are much more robust. They can handle a couple extra minutes relative to the hammerhead shark. So we make those calls in the moment depending on what we get. And then also, of course, we want to double check what we're doing, right? So not only, can you blood sample for just fisheries work, But any shark that's coming up, you can get a blood sample to kind of check your own influence on the stress physiology of the animal. So something that we often do is take a blood sample right when the shark comes up on board. And that's basically going to be associated with the capture stress. And then if everything's clicking and working well and we have time, we'll get a blood sample right before release. So it allows us to also look at any of exacerbations that may occur while they are on the cradle, while we're working them up. And we respond accordingly to that data. So yeah, we do try to be mindful.
I've got a couple of questions online that I want to address real quick, and then we'll get back
Okay.
to the audience. So this comes from John. Does the research being done in aquariums such as the Georgia Aquarium provide any data that helps this research?
Yeah, there is actually definitely overlap. So obviously veterinarian staff in aquariums are, you know, very, very aware of the physiological pathways and all of that, and are monitoring the health of their own captive animals. And I would say that there's not always collaborative research between, shark scientists and vet staff, not for any bad reason, just because we're kind of doing different things. But there are definitely, I would say, an increasing amount of conversation and collaboration with vet staff. So myself, Jake, other colleagues have definitely worked with veterinarians. And it's actually really wonderful to have those conversations because we kind of have this way of thinking about it. And then they have this other kind of set of ideas. And when you start to bring them together, it definitely raises some interesting questions and helps to kind of mature the questions that we're looking at, the methods we're using and stuff like that. So I would say it's not happening all the time, but when it happens, it's really great and definitely is something that I would personally like to do more.
Awesome. One other question that it's Maggie.
Maggie.
So great talk, Heather. What do you expect to see from the microbiome might be indicative of shark immune stress?
Oh, interesting.
Yeah. This was not a
Tough one,
pre-prepared
Maggie.
question.
Yeah. Yeah, so the thinking with the microbiome study is, first just kind of assessing potential different communities of microbes that live in different areas across the shark's body. So when we collect our microbiome samples, we're actually sampling on different locations on the shark. And for those of you who have learned about sharks, perhaps, you know that the immune system of sharks are kind of notoriously quite robust. There used to be kind of a misnomer that sharks didn't get cancer. That's not entirely true, but they do have a very low incidence of cancer, low incidence of disease, high ability to heal from wounds, stuff like that. So they have a pretty robust immune system. And what could potentially be very interesting is that these microbiome communities are potentially helping out with that immunity that they have. And potentially helping out with, stress response and different anthropogenic stressors and stuff like that. So I would say at this point, we're just kind of starting to chip away at the beginning of these questions. There are shark microbiome projects that have been studied, but, we're trying to get more species and kind of just get an idea of what these communities are. And then we can start making those kind of connections to, does this help immunity? Does this help with stress response? Is a stress response throwing off their microbiomes that are so important, right, at the time of capture? When we are doing microbiome sampling, we are super careful to not sample where we're also holding the animal, right, because that'll throw off our data. So if sharks are losing those microbiome communities in a capture process, does that impact their ability to recover? So definitely some interesting questions there. Thank you, Meg.
So we've kept Heather for well over an hour. I want just maybe time for one or maybe two more questions.
Sure.
And then we'll
Yeah.
end. Saw
Sounds good.
Hans got a question for here.
Hi, thank you for your talk. I'm Ron, one of the surges. I had a question. So, earlier you talked about saying that there are some other theories about how
Mhm.
interspecific differences between species shape their vulnerability,
Mhm.
combining physiological data and other data. Could you elaborate a little bit more what those theories are and if there are any ongoing attempts to study it? Because it seemed like a really interesting like
Yeah.
subject for, I mean, just for me, like that difference of vulnerability is so high.
Yeah, absolutely. Yeah, so the behavior aspect of it, I think, is probably a big part of it for certain species. So when a shark is captured on the line, some species just kind of go crazy. Like they just are thrashing around, they're going nuts. They're just like really active on the line. So that would be something like a black tip shark. If any of you have done recreational fishing, they just come flying out of the water. But other species, like a tiger shark, right, they're much calmer. They may not necessarily have the same kind of like extreme behavioral response to the capture, in which case, if you're just thrashing around a lot, you are using those muscles, you've got contraction turnover, you're producing lactic acid, you're driving up those values, and it may just get them to a physiological disruption quicker than other species. There is also an aspect of it, which is some species have to swim in order to breathe. Some can sit on the bottom and open and close their mouth, which is called buccal pumping, and respire that way. So the species that have the ability to open and close their mouth and respire may be able to do that while they're still captured, in which case they're bringing in oxygen and they're helping with recovery. Whereas those that have to swim in order to breathe may be at a disadvantage in that case. So that's kind of the behavioral side of it. And people are doing studies to look at that. So there are some interesting ideas in terms of putting out fishing lines and close to the hook, having accelerometers or having camera tags or something like that, where you're documenting the behavior of the animals at the time of capture. Physiologically speaking, there's a lot of interesting questions. So it could just be an innate physiological difference, which at this time we definitely don't really understand why. But I will say, like for dusky sharks, they just get to that physiological upset so much quicker than those sandbar sharks. When they come up online, their muscles are all locked up, and that's going to be a physiologically driven thing. And if their muscles are locked up, when you throw them back overboard, they're not going to be able to swim, in which case then they can't respire and recover. The physiology of it is super interesting, but we don't know it. There are other theories about, in terms of studying that. So like studying maybe parameter influence on the heart or parameter influence on locomotor muscles, that's going to be a lot of lab work. So this is where physiology kind of carries over into lab intensive projects, which definitely I've collaborated on one of these projects, and there are those sorts of work happening. Other theories are like with the hammerhead sharks. There was one paper that was put out that said their mouths are very small, and maybe that's why they're kind of at a higher disadvantage. So, more data remains to be shown around that. So yeah, those are just some examples. I could go a little bit longer, but I don't want to take too long. But yeah, there's a lot of interesting questions for sure.
Well, Heather, thank you so much for giving us a wonderful seminar tonight.
Thanks for having me. Thank you.
For those of you that are still on campus or on campus for the rest of this week, Heather will also be here. So please catch her at a mealtime or some other opportunity if you have other questions. So thank you. It's a wonderful, wonderful seminar. For those of you online, I hope that you'll please join us at 8 o'clock again next Tuesday for our next seminar series. And otherwise, thank you very much for coming, and good night.